We really want to work as a community together to be able to combat this outbreak as it's happening. My lab have been working in Sierra Leone and Nigeria for a very long time, working on another virus called Lassa virus that's similar in that it causes hemorrhagic fever like Ebola. And when the outbreak began in West Africa, we helped our teams there set up diagnosis and surveillance on ground.
I was involved in the diagnosis of the first Ebola case in Nigeria. And the reason why we were ready with the tools and then the reagent for diagnosing Ebola is because through my collaborators at Harvard, I'm involved in the work that is going on in Sierra Leone. And when this particular patient presented with clinical signs, symptoms similar to Ebola, and the sample were collected, naturally they thought of sending the sample to our labs.
We sequenced 99 samples from 78 patients that were the first patients diagnosed in Sierra Leone as part of the Ebola outbreak. And in fact, we captured 94% of patients diagnosed in Sierra Leone in the first 3 weeks of the infection. And the kinds of things that we could figure out from it is we could see that if you look at the 3 published Ghanaian strains, that these are all linked. These are all appear to be one chain of events. And particularly, we can really have a close view of what happened in Sierra Leone. And it seems like there was one entry into Sierra Leone that then spread throughout. We see hundreds of mutations, over 300 mutations that separate this virus from past viruses. We see about 55 mutations that have just happened within the course of this outbreak event. And we also see hundreds of mutations that are happening within individuals, things that may one day become common in the population and fixed between individuals. Many of those are functional, so we're going to be paying attention to those.
This is providing information live, and for people that are interested in going forward, developing either drugs or vaccines or having understanding of pathogenesis, that actually provide, crucial information for people to go on and then develop whatever tools.
A lot of the diagnostics are built on the genome sequence of the virus itself, so we can say which ones are would still be sensitive and which ones the virus is sort of mutated away from. Same thing with vaccines. Vaccines are built on the protein sequence, which are themselves built on the genome sequence. So we can see if that's changing. There are certain important proteins that are critical to that, and as well with the drugs as well. So it's the type of genome sequencing we do informs whether or not the drugs, the vaccines, the diagnostics that we're using are going to be viable as the virus continues to mutate and change.